Much for the opportunity to speak, and I'm really sorry for not being there in person. I'm truly delighted to be part of this wonderful meeting, and I've already learned a lot. So, what I'm going to talk about is our work on AI for healthcare, genomic medicine, and therapeutic development. So, there's multiple facets of our lab. On one hand, we're trying to understand the path between genomics to circuitry, understanding targets, being able to design drugs and then eventually test their affinity and predict their affinity in both animals and patients. Second, for existing drugs, we're trying to understand patient heterogeneity, trajectories of patients, understanding how we can repurpose drugs and how we can understand multimodal representations of patients across both text and, of course, structured information. And then lastly, we're interested in foundational AI models spanning the whole spectrum of all of these different types of data sets, and in particular in drug discovery, in protein function, and in patient states. Medicine has really come a long way since the time of Hippocrates in ancient Greece, of basically looking at patients from the outside and then making some kind of interventions that were very often wrong and very often harmed patients. The major transition happened with, of course, the Renaissance and the advance of new technologies for being able to probe inside the body, being able to recognize individual components that were pathogenic. And as early as 115 years ago, Alois Alzheimer's diagnosed the first patient with Alzheimer's disease using subcellular phenotypes, being able to see inside the cells and recognize the amyloid plaques and the neurofibrillary tangles that you can see here in this picture. So this is looking at 4 cells and 2 phenotypes. By contrast, where we are today is from our own work published in Nature last year, we have 2.4 million cells across 430 donors. And every single one of those dots represents one of these cells. But now instead of measuring 2 phenotypes, we're measuring 20,000 phenotypes. So the expression of every single gene. So that's where, of course, these foundation models that we're talking about come in. And that's where understanding representations of individual cells, of patients, of individual genes, and these circuits is extremely important. And the other dramatic advance is, of course, the ability to not only look at correlations with expression information and molecular and cellular pathology, but also causation by looking at genetic variants and their contributions to complex traits. In this particular case, you can see ApoE, the strongest association with Alzheimer's disease, basically reigning supreme among all the other ones. But we now have thousands of genetic associations across many different facets of each of these major disorders, more than 120,000 such variants. So there's 3 major paradigm shifts that I would say have been resulting from these technological advances. The first is, of course, going from hypothesis-driven research to data-driven research. Instead of first formulating the hypothesis and then gathering the data, doing all of the thinking before, and then having very targeted studies, we're now able to gather massive amounts of data and systematic data sets, building these resources with massive data sharing of both data sets and models, and having these comprehensive views where you can basically shoot first and ask questions later. The second one is going from correlation to causation, not just correlating epidemiological variables with outcomes, but also thinking about the impact of, number 1, individual genetic variants, number 2, understanding polygenic scores to reveal causal factors through Mendelian randomization and other approaches, and number 3, of course, large-scale perturbation experiments that allow us to now confirm these hypothesized causalities. But I think what this group is really focusing on today is the dramatic transition from classical data analysis to generative AI. And instead of having a new methodology for every problem where the human scientist does all the thinking with very few parameters and targeted models, being able to look at these foundation models that are really multimodal, that are representing the data in completely new ways, and that are hierarchical and truly understanding of the concepts. So that's the excitement, and that's sort of the what I would call the modern deep learning, basically being able to not only make predictive functions that look at an image and then make and give an output, but also being able to systematically learn representations where the computation should be done. In other words, instead of computing directly on the pixels, that basically proved insufficient for many decades, being able to compute on a latent space representation. And that's what the major advance of many of these foundation models allows us to do. And we've been paying a lot of attention to this latent space and sort of creating tools for directly manipulating these latent spaces across many different data modalities. So for images, the X, the data are just the individual pixels, the Y are the prediction, and then the latent representations are lines and shapes and components of a person's face or a wheel of a car and so on and so forth. So these are the primitives that are being learned at increasingly abstract levels. For DNA sequences, these primitives are basically the motifs that are recognized and able to be reused and combined in higher levels of abstractions. For chemistry, and protein structure becomes even more interesting because there isn't a simple grid, but instead it's very much a graph where the representation of any one atom is, fairly low information content. But when you combine multiple atoms together, then you have emergent properties. So at every layer of abstraction, you basically can combine more and more of these properties. And of course, when you look at entire patient records, you can now have representations, in this particular case, looking at the major axis of variation of 400 plus Alzheimer's patients, revealing an axis of cognition along this diagonal and the axis of pathology along another diagonal. But of course, the major advance in the last few years has been in combining all of these representations also with natural language, being able to represent the latent representations, the embeddings of individual words or entire scientific papers or the descriptions associated with a protein function. And of course, through the work of Marinka Zitnik, our collaborator, and many others, we now have the ability to now have knowledge graphs contributing to these representations, where similar to these individual atoms receiving information from their neighbors, every one of these genes, every one of these cells, every one of these patients can be connected to each other, and those latent representations can basically influence each other. In other words, if you have a gene that is associated with a lot of lipid functions, that gene becomes more lipid-like. And if Alzheimer's is now associated with those genes, then Alzheimer's itself becomes more lipid-like as the genes become more Alzheimer's-like, and the lipid functions become more Alzheimer's-like as well. So we're using these representations to now start influencing each other and revising the latent representations for each of these different data modalities. So that allows us, of course, to look at pictures, as I showed in the first slide, with these cells where now the quantitative variables, the quantitative vectors of 20,000 dimensions can be interpreted in a much more meaningful lower dimensional space. And of course, two dimensions is very limiting, but working in 20 dimensions or 50 dimensions for gene expression is actually, in my view, much more meaningful than in 20,000 dimensions. And those dimensions now can have what we call highway names and street names that understand both the global variation and the local axis of variation. And then lastly, the multimodality is what allows us to now couple interpretability to each of those. For example, you can co-embed chemical structures along with patents that describe the function of those chemicals. And that has allowed us to now start understanding regions of the chemical space that are fulfilling specific functions. So this multimodality brings in interpretability. So this is one example where my student Emma Tissinger basically carried out this study that combined the prime PG that Marinka's group developed with an additional network that is able to now learn the structural representation of each of these proteins, that is able to now build a joint representation across both knowledge graphs and structural information. So you can basically now have both of them together. And this allows you to now start understanding the latent spaces of the proteins, of course, but in the context of all of the additional information. So every dot here represents one of the entities in PRIME-KG. And here within the gene and protein space, you can see the individual functions emerging. So what's really exciting is that we can now interact directly with these spaces. So we have built a tool that we call Mantis for basically being able to directly visualize and ask questions about these latent spaces and being able to, of course, use agentic workflows, as I'll describe later, to be able to really gain insights and probe these directly. So what does that allow us to do? What does that allow us to do now? That allows us to now start building for each of these different data modalities, latent representations that can be queried, that can be reshaped based on these interactions, and that can allow us to take, for example, single cell expression and enrich it with the descriptions of the functions of these corresponding proteins that are being expressed. So instead of thinking of SCGPT as gathering just simply numbers that are meaningless from gene IDs that themselves carry zero information, you can now start enriching these representations for the latent spaces of cells using the more functional view of the corresponding proteins. What that allows us to do now is also start understanding the relationships between drug embeddings, where we can basically now take the chemical representation, the graph representation of every one of these molecules, and combine them with the much more information-rich representation of all of these proteins. So you can now start looking at the linking between two floors, if you wish, in these latent spaces. One floor is representing the chemicals, and another floor is representing the proteins. And of course, you can now spread information back and forth. You can build joint representations that allow you to now understand the binding affinity of different proteins based on where they are in this latent space. And of course, this doesn't just stop between proteins and drugs. You can also look at the targets themselves. You can look at the clinical subjects. You can look at patents and FDA filings within the same regions. And therefore, you can build these knowledge graphs that allow you to flow much more meaningful information across all of them. So one application of this has been building this affinity net model that basically seeks to carry this out in the space of chemistry, where you can basically build these drug fingerprints that are using these 20,000 dimensional representations. You can build protein embeddings from Prot T5 in 1,000 dimensional representations. You can also embed the domain descriptions using PubMedBird. And you can also embed the patents, as I mentioned earlier, also represented using this BERT model. So you can now combine the affinity regression as well as contrastive alignment of drug and protein embeddings and understand the ability to not only embed these, but also outbed them. In other words, the ability to create chemicals that are evaluated based on the reconstruction of the original fingerprints for these molecules. Why is this important? Because it forces the latent representation to actually capture the full information of those chemicals. And we found that when you include this reconstruction ability, you don't need to effectively start thinking about a separate model, like a diffusion model or other generative models that you can then couple to be able to create new molecules. By forcing the model to have this outbedding capability, we can now sample in chemical space and basically directly recognize drugs and molecules that allow us to now directly oversample, basically generate molecules from very narrow regions of the space that allows us to now start generating new drugs. So we basically have these multiple loss functions, basically not only the reconstruction, but also the ability to directly predict the affinity. So we have this query key component that allows us to look at the chemical projection as well as the protein projection and the domain projection, where the value is the actual predicted affinity. So when we put all of this together, we're basically able to now, in a very rapid fashion, because these models are, carrying these out in the entire protein landscape can be now performed in the order of minutes, where you can basically now build these combined representations. We can now start looking at where are the chemicals in this giant landscape that are associated with specific biological functions. And where we can also take experimental information and map it onto these machine learning predicted affinity landscapes. So this is, for example, the latent space of a particular drug screen for LMP2. And that allows us to now recognize that, in fact, the experimental values that you show in these circles are, in fact, matching the machine learning inferred values. And then we see that there's actually smoothness in the space, that the further away we get, the lower the affinity, and also that there's actually clustering, that the latent space captures the experimental IC50 of these molecules. And then here you can see this ability to now create outbeddings, where I can basically go in different regions of the space that are associated with particular functions that I care about, and actually generate meaningful molecules that are having affinity in that space. And that allows us to now start combining all of this information together in these agentic workflows that allows us to now start predicting, for example, the molecules themselves. So if I manage to go back here, there you go. So basically, we can now create these agents that are able to synthesize molecules, to sort of generate molecules, to sort of invent new molecules that have affinities that are unthinkable for models that are purely machine learning based. So instead of having a biophysical simulation, these models are directly generated from these latent embeddings. They're sampled from that underlying distribution, even though they've never been observed before. And these molecules now have biophysical predicted affinities that are truly off scale. So we can now couple this component of the drug generation together in these agentic drug discovery pipelines with all of the other data sets that our lab has been generating for many years. So basically, this allows us to now start describing, for example, if you want to find a combination of approved post-phase 1 clinical trial drugs that can treat Alzheimer's, there's an expression agent that says, okay, I'll begin by searching for cell type specific mechanisms. Then I will use a tool, because we have provided tools for each of these agents, to both keep them grounded and to sort of give them this ability to sort of navigate giant corpuses without getting confused. So you can now basically search the corpus based on the literature. It says, okay, this particular protein could reduce tau pathology. The targets must be differentially expressed. So we're going to use differential expression, bringing in all of our single cell expression information in the human brain, and then identifying two genes. You can then look at the target validation, check the modules from our expression data set using the co-expression patterns of individual genes across many cells to define these modules, and then checking the tissue expression, identifying biomarkers, and then now you can now bring in this molecule generation approach and then select small molecules by sampling the underlying chemical landscape and then evaluating them by docking each of the proteins. So you can now see these agents in action here, where you can basically see how they're reasoning, basically what is the tool that they're calling, how are they thinking about this. When they're searching the corpus, for example, they're like reviewing each of these molecules and then selecting the drugs, validating the targets, and of course, generating a summary report based on all of the information together. So that allows us to now start building these massive models. So instead of 50 or 100 or 1,000, 10,000 drugs, we're basically now looking at 121 million chemicals that we have encoded using AffinityNet. That allows us to now start understanding those in the context of proteins, diseases, and drugs. We can basically see which proteins interact with each other, which are near each other, which are implicated in what diseases, and which drugs are both interacting and implicated. So now, of course, in collaboration with Marinka and Brad Pentelute, we're now trying to integrate all of this information in being able to reason about, of course, language as well as biological information, these single cell models, and of course, all of the geometric deep learning that Marinka's group is really leading to now start building these complete agentic workflows for reasoning over the molecules, the targets, the patients, and start predicting individual drugs for Alzheimer's, metastatic melanoma, cardiovascular disease, and so on and so forth. So this is all at the level of proteins and drugs. We're also doing this kind of work in the level of gene expression patterns. And in particular, a lot of the traditional views for single cell expression have made the simplifying assumption that every cell from an Alzheimer's patient is an Alzheimer's cell, and every cell from a control patient is a control cell. That assumption is always implicit in every one of those differential expression methods. So what we can do instead is start looking at neighborhoods of cells where we can now project the phenotypes of the corresponding donors that these cells were sampled from, and then use that to now recognize Alzheimer's enriched cellular neighborhoods and control enriched cellular neighborhoods, where basically all of the cells there, or at least the vast majority of the cells, are coming from control donors, and here from risk donors, in red for risk and blue for controls. So that allows us to now start reevaluating the phenotype of each cell based on the neighborhood that it belongs in, rather than the binary Alzheimer's, non-Alzheimer's phenotype of the donor. So that allows us to now recognize an entire gradient of pathogenicity for every cell, where we can now look at the 326 donors. These are the original donor phenotypes. But after you go through the neighborhood exercise, you can basically see that there's a score for every one of the cells. And that allows you to now have a quantitative readout of every cell based on how Alzheimer's-like it is based on the neighborhood scores, and therefore go back to the donors and say, well, the cells from this donor, even though that donor was, I don't know, red, were all sitting in blue neighborhoods. And therefore, the microglia-specific score for that donor is going to be blue, even though that donor is actually Alzheimer's diagnosed donor, their microglia appear to be healthy. And of course, when we look at other cell types, we see that that's where the pathology is. So we can do this for every one of the different cell types associated with every one of these donors across dozens of cell types. And now what we can find is that some donors have very high scores for Alzheimer's in their astrocytes, but very low scores in, say, their excitatory neurons. And that allows us to now start predict not just for Alzheimer's as a single phenotype, but in fact for tangles and cognitive impairment and tau pathology and neuroinflammation, for every one of those pathologies, for every one of those cells and for every one of those donors, and for every one of the cell types, we can now have a much more quantitative readout. Why is that important? Because if you look at the differential expression abilities now to recognize differentially expressed genes, you see that we could previously recognize 4 or 5 genes. We can now recognize hundreds of genes. And in fact, those are the same genes, but just dramatically more significant. So it's not like we're making up data, we're simply enriching the signal that is already there. And that allows us to now start recognizing phenotypic subtypes of donors, transcriptional subtypes, and how they relate to each other. So that allows us to now start recognizing the hallmarks of Alzheimer's disease and how they're manifesting at the gene level, which allows us to now start selecting target genes that we should be developing these drugs for, in specific cell types for specific subtypes of donors. So we can now start relating the donor phenotypes with the transcriptional phenotypes. And of course, we can use our capabilities again to basically start studying the cartography of patients as well. So this is now looking at mimic patients, for example, and how you can now use these models to now start recognizing different subtypes of patients. So of course, our goal ultimately is to be able to carry out these types of studies. So this is an example that we published in the New England Journal of Medicine a few years ago that basically is able to now take the phenotype of obesity and then break it down into very specific mechanistic basis that you can then manipulate to reverse the disease phenotype at the cellular level and of course at the organismal level and of course at the epigenomic level, at the regulator level, the motif level, the enhancer level, the downstream target genes, and all of the cellular and molecular phenotypes. So that's where I want to sort of wrap my talk by basically saying that it's really important to have this transparency through the direct visualization of these latent spaces. Namely, we've made extraordinary advances in AI with deep representational learning, this multimodal understanding, this intuitive reasoning. But I feel that a place where we took a wrong turn is that we're pushing more and more language-based interactions where the AI is a black box and it does all of this amazing thinking, but it doesn't allow us to peek inside its brain, and the humans are left out of the loop. So that's why we developed this tool Mantis that I alluded to, and I showed you just a very basic functionality of being able to browse. But we have, of course, these ontologies that are discovered automatically, where you can basically now focus on specific classes of proteins, on specific types. You can now, of course, search. You can use, the search function here to basically look at, for example, all lipid proteins or you name it. You have the ability to search, of course, and see the results inside these neighborhoods. And of course, we have all of these agents and an orchestrator where when you're asking natural language questions, it's able to sometimes write code that it will then run, plot information, or answer in line. So these are some of the expanded capabilities here of basically being able to sort of recognize patterns, which in text are keywords, but in proteins become protein domains, being able to sort of see this multimodal information, being able to see the actual, and being able to collaborate with other partners. And then that allows you to sort of, for example, browse millions of proteins or millions of articles and then discover these ontologies directly. And through the use of these floors that I mentioned earlier, you can basically see how drugs relate to proteins, in this particular case, how papers relate to grants and to individual offices, how knowledge changes over time, how quantitative information can be mapped onto these, and then, of course, links. And all of this information is multimodal. So we can now look at images in the latent landscape. We can look at protein structures. We can look at chemical structures. We can look at all of this information natively within this. And that's what I think the power of maps is, the ability to know not only take a left turn, take a right turn through text interventions of the AI guiding us, but instead by directly visualizing the abstractions, summarizing, being able to guide decision making. So these are the four pillars of Mantis, basically on one hand having the embedding, on the other hand the knowledge graphs, the agents, and the tools. And these are all of the different components. So we're very excited to collaborate with people who want to study their own machine learning models through this ability and to be able to apply this in many different landscapes. So for example, we can use that for chatting and visualizing and asking questions to the agents, being able to build all of these data science primitives of bags and trajectories and partitions and trees and categories, and then interacting with all of those directly. Being able to carry out quantitative data analysis directly through the landscape, being able to see this information in 3D or in 2D representations, being able to collaborate with other humans by launching these queries that involve sometimes multiple people co-browsing the same landscape or leaving asynchronous nodes that are geotagged, or being able to request more complex queries that require coding. This is an example of the agents not only carrying out data analysis in the back end, but also creating visual interfaces in the front end that allow you to now interact with the resulting capabilities in an interactive fashion. And then the whole platform is embeddable inside other tools. For example, we have a Chrome extension that allows you to, for example, browse Wikipedia through the latent landscapes of this information, then directly click on multiple articles and then see their similarities. And then this is now using these workflows to develop new drugs, where you can basically now create these, PubMed agents and Google Sheet agents and orchestrator agents, and then you can now create these workflows that allow you to carry out all of your data analysis needs. And then lastly, of course, we're using these to coordinate our own team, where we can basically study the information from our own GitHub or our own conversations and meeting recordings. So that's my vision for where the road is ahead, basically this systematic understanding of both the biological landscape through these embeddings and these visualizations, but also our own work and being able to use AI as a Google map for navigating these knowledge landscapes. So being able to sort of have this F18 dashboard that allows you to sort of understand all of these different modalities of the data. So that's where I'll stop and thank both the computational biology component of our lab, as well as this new Mantis component of our lab. And I'll be delighted to take any questions.
Thanks, Manolis, for the wonderful talk. Any questions?
Yes, thanks from my side as well. Really cool how you build embeddings and explore them. And I was curious on that regard, on your agent-based discovery of drugs, how you configure the agents so that they explore your embeddings. Like, is there a specific set of vocabulary or interaction capabilities they have, and what's your vision there as well?
Yeah, it's a fantastic question. So basically what we have done is that we have created this MCP framework across the entire toolkits of Mantis. So basically, if you look here, we have these agentic workflows and we have these MCP servers where every one of the core capabilities of Mantis, like point selection, trees, gradients, search, partitions, iterators, etc., these are all encapsulated in agentic workflows as MCP registered workers, if you wish, so that the orchestrator is able to now reason about each of those components and is able to now call them and interact both with the front end and the back end. So we have written tools that take the code itself of each of these procedures and then describe them in English text, which is then used to dictate the capabilities of each of those processes. And therefore, as a developer, you only need to create one of those capabilities. And we have this marketplace where you can basically have these third-party tools that are developed that are able to reason and use all of these different capabilities that are now registerable and usable. So you can the agents are able to reason over the embeddings themselves through these core capabilities, the knowledge graphs and the knowledge extraction through these graph neural networks, all of these entity recognition, relationship extraction, and of course utilize the tools that we have for this programmatic automation. I hope that answers your question. Thank you.
Professor Kellis, thank you for the wonderful talk. I wanted to ask, when you're using embeddings for your data, how do you measure like information loss when you put embeddings versus process the data entirely, et cetera? That's kind of been a challenge with dealing with some of these data sets, where trying to balance how much information you lose with an embedding versus how much you gain when you have the embedding across different modalities, et cetera.
Yeah, this is a fantastic question. So, as I mentioned earlier, one of our key design principles is the ability to carry out outbeddings, to basically to be able to reconstruct the original document. And we have these outbeddings in the context of chemistry. We also have them in the context of natural language. So, we have now the vector representation of text, and we're training these embeddings and fine-tuning these embeddings to be able to now accurately reconstruct the text. But what I'm actually much more excited about is this tunable abstraction, this tunable representation resolution, if you wish. And the tunable part is what I'm personally the most excited about. The fact that I can choose if I want to think of a molecule as that exact molecule and nothing else, or the larger class and the larger class and the larger class. And I think that's where it's two sides of the same coin. In other words, we can't have creativity and perfect accuracy at the same time. Like this ability to abstract away from this one example to a class of elements is fundamental to everything we're doing. And in Mantis itself, what we have found is that the ability to now embed not just individual points, but also the description of the cluster hierarchy that the point is part of, I think is facilitating the representation and the interpretation of the data by the agents. In other words, by understanding a corpus at a time, not just the whole universe of information, in planet Earth, but like a corpus of molecules associated with Alzheimer's, or a corpus of patents associated with cardiovascular disease, etc. Within that corpus, every single one of the elements has a series of clusters that it's part of hierarchically. By appending the name of the cluster along with the element, we're finding that the reasoning actually becomes much cleaner. So this is, this suggests that this, again, multi-resolution tunable representation is helping the agents themselves reason. I hope that answers your question.
More later when I'm back on campus. Thank you.
Yeah, super excited to hear. Yeah.
Hi, and thank you for the fantastic talk. I'm actually a quick question about the last point. It's very interesting to see how you make the these different modalities become navigable, right, through these visual tools. I also very much agree with the notion that there's maybe too much. It's a good illustration, right? That there's maybe too much work that's language centric. But language models, because of all the resources, right, that have been poured into them, because of the ability to get literature, get us at a lot of things: reasoning, test time scaling, interpretability. So I'm wondering how you're thinking of maybe a more virtuous, more effective balance between the text world, the language world, and these non-language modalities and embeddings.
Yeah, absolutely. It's a fantastic question. And it's a point of clarification that I want to make. Basically, when I said that the field took a wrong turn, I did not mean that we use language everywhere. I think that's a fantastic thing. What I mean is that we are happy when the model just simply tells us, here's the answer. What I'm saying is not stop using language as the glue. I think that for humans, language is the glue. That's how we think. And that's why I was describing in my early slide here how the multimodality, I think, is an important thing, and the language is absolutely the glue. All I'm saying is that when we interact with these models, even when you're just interacting with New York Times articles, if you're just interacting with like 3 million New York Times articles, if your only modality is text, if you don't have that visual representation, then you're not able to get the bigger picture. I think that a picture is worth 1,000 words. And in this particular case, a picture is worth 1,000 documents. So by understanding that landscape, this is a text-only example, but it's still visual in the interaction. So basically, the point that I'm trying to make, and thank you for allowing me to clarify it, is that we can't simply interact with a map by receiving a bunch of text messages on your phone as you're skiing down the slope. Turn left, turn right, turn left. And that's what the language-only interaction would be. The analogy that I like to give is Aladdin falling in the Cave of Wonders and being locked inside a box. And all it can do is ask the genie questions in a piece of paper. And what we've built is a magic carpet that allows Aladdin to sort of fly and see the data directly. So that's where I was basically saying that the visual component is extremely important. I do agree that language co-embeddings are crucial. And we've done some very exciting work with Marinka on Procyon, for example, that has both a language and protein structure co-embedding of proteins. And I think that the language component with chemistry, as I showed you with the patents, and with cells, as I showed you with the richer descriptions of protein functions when we're doing our cellular embeddings, all of these things are extremely important. But we as humans have a tiny language center in our brain and a giant visual cortex. And I think that engaging our visual cortex is probably going to be the key to humans feeling that they're not left out. And I think that by bringing the humans back, everybody has to gain. AI has to gain, science has to gain, transparency, interpretability, trustworthiness, all of these things that we worry so much about. A big chunk of the worry is because we feel left out. We don't see the data.
Thank you so much.
Let's thank Manolis again. Now,
thank you, guys. Sorry for not being there in person, but excited to hear all of your
Thank you, Manolis. Yeah. Now we are going to start the poster session. We are going to come back at 12 p.m. For Professor Yuzui's talk.